Okay, so our first speaker today is Anthony, and I've asked him, I don't know, 10, not quite 10 times how to properly pronounce his last name, and I already blanked it. Yannarell. I got it, Yannarell. Anthony Yannarell from the University of Illinois, and which is a long ways from here, so hopefully he's over the little bit of jet lag. And he's been studying microbial communities in water, sediment, and soils for about 15 years, so he's got a lot of experience in this area. And he's really seeking to understand the interactions between the microorganisms, the plant, and the soil environment. He's got a great topic to share with us today, essentially looking at how the soil microbes are affecting weeds and weed suppression. So I can't wait to hear about it, and I'm just going to let you get going.
All right. Well, thank you very much, Heather, and thank you guys for coming this morning. I'm glad you did the introduction because I realized that in my first couple slides there's absolutely nothing about me being a microbial ecologist, so I didn't want to surprise everybody when I start talking about microbes. But yeah, so what I'm going to be talking about is plant-soil microbe interactions, very early stage research, and things that might be interesting in terms of weed suppression potential in agricultural systems. And so I'm a soil microbial ecologist who's been gradually getting more and more interested in agricultural systems. I give a lot of the credit to dragging my interest towards ag to this young woman right here, my former graduate student, E. Liu, who graduated with her PhD just this past December. She's really the one who got this project off the ground and really got me interested in weed suppression and the potential microbial role inside that field. So I've had E's, whenever I say we, really I mean E, I mean her. She's the one who did almost all the work here, and I'm just up here showing this to you. And so I want to acknowledge her role in this, as long as the role of a lot of my collaborators and assistants, some of the farmers who worked with us on this project. And then, of course, funding from this is coming from a research grant from the USDA NIFA and also from the Ceres Trust Organic Research Foundation. I like to start off showing this slide. It's one of my favorite pictures I've snapped. This is from an experiment that we do out on the South Campus in the University of Illinois. It's actually a tillage and cover crop experiment, so it's combined treatment with tillage and cover crops. I took this picture 2 years ago in the spring. You can see some of our different plots here. And, you know, I don't want to get into the details of this particular experiment, but one of the things you're looking at is some of these plots had a rye cover crop, ryegrass cover crop, and some of those were in our no cover crop control treatments here. And all the green you can see in this picture right here is giant foxtail. We had a weed infestation in the spring of 2014, really severe. They had to come out and spray again. But notice that the weeds are not distributed equally across this picture. This picture right here in the middle that has the lowest coverage of weeds, that was one of our rye cover crop treatments. And in the back, a couple over, you can see what appears to be another bare spot. That also was a rye cover crop treatment. And so this rye cover crop was providing some weed suppression potential in the soil. And the other thing that I love to point out about this is that, okay, there was cover crop and no cover crop in this experiment. The cover crop is gone. So we've actually killed this thing off and plowed in the soil as a green manure a month previous to when I took this picture. So even though the cover crop has not been on the field for a while, it's providing some of this lasting weed suppression into the soil. Interesting thing. Now, it may not be completely unknown to you that if you use cover crops as green manures, they can provide some of this weed suppression potential. And one of the things that we know they do is that when you kill off a plant and use it for green manure, it releases all of its plant guts and chemicals into the soil. And some of those chemicals have phytotoxic anti-weed properties. I'm showing you a particular class up here. These are all isoflavones that are found in a lot of legume cover crops, and these are all known to have phytotoxic effects. So that's something that we know about. But I'm a microbial ecologist, and I know that there are microbes in the soil. Microbes interact with plants, both the living plants and the dead plants, the germinating plants, and that got me and E thinking about this problem a little bit more. One of the reasons I was thinking about this is because one of the centerpieces of my research program is what ecologists call plant-soil feedback theory. And I don't have time to go through the whole theory here, plus it would probably bore you all anyway. So I just want to, boil it down here to this main slide. The idea between plant-soil feedback is that plants influence the soil properties, including soil microbial communities, and those microbes then in turn influence plant performance. They can help or they can hurt plant growth. And when you have a multi-species system, every plant in the system is involved with these interactions with their own microbes. They're all generating feedback to themselves, but more importantly, this plant's microbes may also be influencing the performance of this other plant in the system. And so you have this capacity for plants to interact with each other indirectly through these below-ground changes that they're making in the soil microbial community. And to make this maybe even a little bit more simple, in terms of the particular case that I want to talk to you about here in the agronomic sense, we can boil this down a little bit further. If this is our cover crop, we don't really care much about how the microbes are influencing it, because we know we're just going to kill the thing off and plow it anyways. But it does make changes to microbes that may have lasting legacy effects on the plants that follow that cover crop in the field, be they weeds or your crop. And so does the cover crop influence the feedback dynamics that occur in the plants that follow it? And to bring this back to the kind of allelochemical story I was telling you before, we have cover crop residues being used as green manures. We know they produce allelochemicals that have weed suppression potential. They have the influence on the microbial community that can also affect disease incidence in the crops and the weeds that come after it, and also weed emergence. And then, of course, microbes eat chemicals in the soil, and so how do they affect the allelochemicals? Do they detoxify them, or do they wind up magnifying their toxicity? So to address this kind of question and experimental setup, what E and I did was we collected some soil from the field, and we also grew up some red clover, which we're going to use as our test cover crop. So here's our red clover growing in the field. We harvested these residues and brought them back to do a greenhouse experiment with some soil that we collected separately. And what we did is we established 4 different treatments. We took our soil and we sterilized half of it to kill off all the microbes. And we left the other live so that we're getting all the microbes that were present in the soil when we started the experiment. And then to our sterile soil and our live soil, we added either just sterilized water as a control treatment or red clover residues. So now we have all the combinations of microbes and red clover accounted for in our treatments here. And so the other thing that I want to say, and to kind of relate this back to organics, because here at an organic research symposium, is that we collected our soil from 6 different working farms around the campus of the University of Illinois. 2 of those were using conventional management, 2 of those were using no-till, and 2 of those were using organic management. So they're all cornfields, but they all use 3 different types of management replicated twice for these different soil sources. So we combined all these. To test the weed suppressiveness, we use what I like to call the soil burrito assay. And it's really pretty simple. Basically what you do is you take the soil that you're going to assay and put it on some moist germination paper, like the beans in a burrito. And then you stick on seeds of the species that you want to test. So in this case, we used wild mustard as our experimental weed. And so we put in multiple wild mustard seeds on our soil that we're going to assay. Then you roll them up like a burrito. You stick them in an incubator for 7 days. And after 7 days, you can pull this thing apart, and you can see how many of the seeds germinated, how many are dead. You can score them for disease incidence. You can do all kinds of things with these seeds. And so that's pretty much what we did. We took all these soils and all these various combinations. We made a bunch of burritos, and we made these burritos on a time series after we had combined our red clover residue. So we're mimicking a time series after when you would plow the red clover into the soil. So we did a full set of burrito assays immediately after we combined all of our treatments. We did them 5 days later, 10 days later, 20 days later, 40 days later. So up to a month, over a month after you would have incorporated these cover crops, we wanted to test to see how the weed suppressiveness may change over that time course. And here's what they look like when you take them apart. Some of our seeds sprout and grow quite nicely, and some of them don't. And we can use that as an index of weed suppressiveness. Okay, so here we have the data, and I'll walk you through it a little bit. Each of these panels represents one of those 4 different treatments that I talked to you about a couple slides ago, so the combination of microbes and red clover residues. And what you're seeing plotted on the y-axis here is the percentage of wild mustard weed seeds that germinated inside each of those burrito assays. So the thing to remember is that high numbers are bad, because that means most of the weeds germinated. If we're looking for weed suppressiveness, we want to see low numbers. And in the top left figure here, this is our treatment where we have just our sterilized soil, no microbes, and no red clover residues. What you see is that all of our soils are giving us very high germination. No weed suppressiveness inherent just in the soil in and of itself. When you add red clover residues to that mix, what you can see is that initially you have very high suppressiveness. That is very low weed germination rates, but gradually over time we begin to get more weeds germinating. So the suppressiveness doesn't last, it tends to go away. When you add microbes into that picture, so this treatment on the bottom here, we have both of our red clover residues and the microbes, you get the same kind of shape, it's just that the suppressiveness goes away faster. All right, so when you put microbes in the picture, what they tend to do is they eat allelochemicals, right? So they tend to mess up the weed suppressiveness of your soil. But interestingly, if you just have microbes in the picture with no red clover residues at all, you can see that we have cut the weed germination rate in half and sometimes even more. So the live microbes are in fact doing something weed suppressive. It's just they're doing it in a different way than when you have the residues in the picture. The other thing that I want to point out about this, let me see, I think, good, I have some of this stuff animated. So we have red clover residues suppress weeds for a short period of time. We have microbes that are capable of suppressing weeds. Notice that doesn't change very much. Most of these lines are parallel, so it doesn't change over time. The other thing that you might be able to see is that these different color lines I put on here are the soils that come from underneath those different management regimes. So the red color here, which tends to be on top on all of the graphs, is our soil from no-till. No-till soil fields in this experiment had the least weed suppressive property, and it also tended to go away the fastest in all of these other ones. The one on the bottom, I don't think I have room for any more arrows here, but the green that you see in the bottom, those are our two organic soils. And so organic soils tend to have the most weed suppressive microbes, and they also tend to have the longest lasting weed suppressive effects when you have residues in the picture. Oh, look at that. Okay, so one of the things I wanted to try to do, because we got microbes, we got weeds, or residues, I wanted to try to figure out the separate contributions of each of these parts of our system. And you can do this with our experimental setup with just a little bit of kind of pairwise comparisons here. So if I want to know just how the residues are impacting things, I can control the amount. I can look at this by looking at the amount of weed germination we get in our control treatment compared to the weed germination that we get in the sterile soil that has the red clovers added to it. If I want to figure out just how much microbes are doing, I can just compare the control soil to the soil that has the live microbes but the no red clover residues. And if I want to find out how those microbes and their red clovers are interacting with each other, I have to be a little bit more tricky. But I can figure out the total weed suppressiveness by comparing my control soils to my live soils plus microbes. And then if I take away from that what I've already calculated above, I can determine if the total weed suppressive is greater than the individual contribution of microbes and residues, or if it's less. So are the residues and the microbes cooperating with each other, or do they tend to be fighting with each other? And I'm portraying that this way here. So again, I've got a bunch of animations on here, but basically what you're looking at, this is our calculated microbe-only suppression on the top. This is our calculated residue-only suppression on the bottom, and this is the interaction between the two of them. Please ignore the Martian letters here. I think that happens when the Macintosh doesn't speak very well to the PC. So they're inconsequential to what we're talking about here. This is days after incorporation. What you can see is that we get consistently high microbe-only suppression and consistently high residue-only suppression from our organic farm fields. All right? So we had a lot of high weed suppression in the organic farm fields. We're getting good contributions from both the microbes and the organic and the residues by themselves. No-till, which had the lowest total weed suppression, also has the worst microbes in terms of weed suppression. So the microbe-only weed suppression effect of the no-till soils was the smallest of all the ones that we measured. And the other thing that I want to point out is that in both the conventional system and the no-till system, the residues by themselves become less effective over time, whereas the residues in the organic system provide continuing support on their own for weed suppression. The reason we tend to lose is that this blue stuff here, whenever you figure in the interaction between the microbes and the residues, it's always a bad interaction. So they're always hurting each other. So what you can think of it as microbes are lessening the weed suppressive effect of the residues, probably by degrading the chemicals. The degradation happens really quickly in no-till and conventional, and it just tends to happen at the very, very end in the organic system. So for the organic system, we get pretty strong microbe suppression, a very long-lasting residue suppression, but at the very end, the microbes kind of turn on and start chewing away that weed suppressive capacity of the organic soils. Okay, this won't mean very much, but I'm a microbial ecologist, so I get excited about this kind of stuff. My main bread and butter is I collect samples from the environment, I grind them up, I take the microbial DNA out, and then I apply sequencing technologies to try to figure out which microbes are living in those samples. And what we did in this case is we collected samples from the dead seeds in our burrito assays, the ones that didn't germinate, and we also collected samples from the live seedlings to try to figure out which microbes were present. These are the ones that may have been killing the weeds that we suppressed or responsible for disease or growth suppression in the weeds that did manage to emerge in those. And one of the things that you can see, so these are just colored by different classes of fungi that we could sequence in here. These are really broad, diverse categories. But what you can see right off the bat, I think, is that in our two organic systems, we tend to get very different fungal communities on both the seeds and the emerged seedlings than we get in the organic or in the conventional and the no-till system. So there's definitely differences between the tillage corn and the no-tillage corn, but some of the most different communities we're getting are in the organic system. So there's something about organic management that's very drastically affecting the microbial communities, and as we saw in the previous two slides, that has functional consequences for the weed suppressive that we get in that system. We did the same kind of thing. This is a little bit more complicated, but we compared different treatments with and without residues to try to figure out for each particular microbe that we were able to sequence, does that residue seem to be enhancing its profile in our communities, or is it suppressing its profile in the communities? In other words, is some of that negative interaction between the residues and the microbes due to the residues poisoning the microbes. And so these are all the classes of organisms that we got that had negative interactions. When you add residues, these things all get hurt by them. The ones on the top of the zero line are the ones that get stimulated by the residues. Okay, so we can see that in addition to having these kind of phytotoxic effects against weeds, the red clover residues have some antimicrobial properties that result in changes to the microbial communities. Lots of negative changes in the microbial communities, but also some positive ones. And so we can use these residues as kind of levers into the soil to influence the microbial community and try to figure out if those changes that we make can have positive agronomic impacts in terms of our yield and our weeds. So these are the guys that are responding to red clover residues. Again, one thing that I always think is striking about this slide is that in general, you've got the same colors on top, but this is the organic soil here. We get different microbes responding to the residues in organic soil. So the management, as it was said earlier, management has a huge influence, not just on your carbon equilibrium, but also the community dynamics that are happening in your soil, the consequences for the plants that come afterwards. And good, so I just got the 5-minute flash, so I'm going to end here. These are kind of my conclusions. My conclusions are there's something here to be looked at further. And so the main question that we're going to be pursuing here is we're going to take these kind of functional data that we get about green manure systems, management systems, and the soil microbial communities, and try to figure out if we can optimize these for weed control and other positive agronomic impacts. And I thank you very much for your attention this morning. I'm happy to take questions. Good question. So the question was, when we sterilize the soil to kill microbes, are we also chemically changing the soil itself? So yes, we do know some things chemically that happen when we do our steam sterilization techniques. One of the most profound changes is that when you kill off a bunch of microbes, they pop open. And what they do is they release all the stuff that's inside them. There's a lot of nitrogen and other nutrients inside microbes. And that's a very well-known effect as people who do these kind of soil sterilization studies. We know that we get this pulse of nutrients that come afterwards. So what we did in our experiment is that after we sterilized our soil, we went in and we measured the nitrogen and phosphorus concentrations in that sterilized soil, and we added back to the unsterilized soil to try to counter that effect. So did we look to see what's predominant? Yeah, that would be a different talk, but we use these kind of DNA sequencing techniques. So I showed you some of these slides here at the class level, but we actually go down to the species level, and I've got these giant lists of microbes. That's one of the things that E and I are doing right now, is we're combing through those species lists to try to figure out particular species that are responsive to our treatments. Mm-hmm. Yeah, so she posed both the question and the answer. Very good. So she knows that this blue is both positively and negatively responding, and that's because this graph was conducted at the species level. So there are some species inside this class, Sodariomycetes, that are negatively impacted, and there are some that are positively impacted. Yes. So the question is, why was no-till not very weed suppressive? I'll be the typical academic and say I don't know, but I can speculate. So when we collected this soil, we collected down to a couple centimeters deep, and in a no-till system, if I would guess where the residue responsive microbes would be, they'd be right in the very top. So it's possible that by collecting soil from deeper in the profile, we kind of diluted out that pool of plant responding microbes, but like I said, that's just a speculation. Yeah, so this is so related as why is the organic so good. That I'll be a little bit cagier about. So actually, my grad student E has a hypothesis, and I'm not so sure I believe it, but it would be worth tracking down. Her hypothesis is that because of the organic management, with its attention to recycling organic matter, that the things that the farmers have been doing to that have actually enlisted an army of microbes that are poised to respond to these kind of residue treatments. So they're good at degrading, they're good at turning things around, they're good at kind of dealing with the diversity of chemical substrates that they're seeing because of the management. So maybe.
So we'll take one more while I...
Right, so the question was, how did we kill our cover crop? So for this particular experiment, we didn't have to worry about burning down the cover crop. We had a field of red clover growing in a completely separate farm. It was different from the ones that we collected the soil. We hand harvested the clover, brought it back in bags, and then we just air dried it and used it that way. But yeah, definitely the killing of it in a real agronomic system would make a big difference, and could definitely be a neat thing to look at.
Great. Thank you.